I would have to agree with that. I don't know who stood up first, but I'm going to go to the top first just because the top has been neglected today.
Thank you. Patty Brennan, University of Wisconsin-Madison. I am a medical informaticist, and my research is on how we help patients understand what's happening to them and use information technologies to do that. And boy, is this a big thing you've got there. So I have a few questions to get started, and some of them may reflect the fact that I'm trained as an engineer and a nurse rather than as a microbiologist. You assured us that there's been very few, almost no safety problems. And so I'd like you to tell us about the few, almost no safety problems that have emerged in the practice so far. Particularly how you're monitoring for them, because the kinds of questions that I would be looking for both the donor and the recipient. Is there a long-term consequence, infectious process? How do we assure any sort of remaining gifts that we hadn't anticipated being given to the individual? I'm thinking here of the DNA transfer back to the mother was a surprise when it was first discovered. Secondly,
why don't we stop with that one and have it answered first? I'll kick it over to Scott.
Sure. Can you just restate the question?
Sure.
I mean, in its simplest form.
How you're monitoring for safety problems and what time interval and what kinds have been discovered so far in terms of the transfer, both for the host and the donor and the recipient.
Right. So, I mean, we have standard procedures for this. Not being a clinical reviewer, I'm not sure that I can cite you chapter and verse, but we have diary cards. We do a fairly thorough job. And when I say we, I mean the sponsor. This is all, and again, each one is different. So a particular trial, depending on how it's being performed and what the endpoint is, they may look at different things.
Okay. So it's explored. There's no standard yet about how far out you monitor for.
Well, that's true, and I think that's one of the aspects that we would like to see long-term follow-up, and that's not always easy to arrange. I think it's safe to say.
And my second question, I think, will be quicker, and that is, is there any return on information to the donor about any discoveries that are found during the evaluation of the contributed material?
Billy Joe.
So not that I know of at this point. Most recommendations in place around genomics itself have just started asking whether or not that's something, and different organizations have put forward recommendations for how they think that should occur. I think there's just simply not enough of an evidence base in place right now to start the return to donor of information. But that's, I'm not entirely sure.
Well, let me call your attention to some work of the Hastings Center from probably 20 years ago, when there was questions about testing tissue samples for HIV, particularly during the circumcision. And there is some discussion about how that needs to be handled, confidentiality, and whether or not it should happen. Thank you.
Great, thank you. I'm going to move over to this gentleman down here.
Ron Germain from NIH. I want to follow up on a more granular level with something related to those questions and a point that Scott made, which is we've been focused on the bacterial microbiome most of the day, even though other things have been mentioned. And viruses wafted their way through your discussion very, very briefly and disappeared. But you analogized to blood transfusion. And it's pretty obvious that not knowing about viruses in blood products has caused major problems until we knew about them and screened them out of the blood supply. And I'm not sure I understand why you would not think that there are virus components, and given Skip Virgin's work and other people's work about chronic viruses, including things that are enteric, that you would not be transferring these and would not capture them in any easy way under these conditions as potential problems. So what am I missing here biologically in terms of thinking that way?
So I just want to clarify, are you talking about in terms of safety concerns with viruses being present? So currently donors are screened for HIV, hepatitis A and B, sometimes for rotavirus. So we do look at that from a safety standpoint as part of standard screening for fecal donors.
Even what?
CMV.
EBV and CMV.
Again, I can't really talk about the details of particular reviews. I would direct you to the College of American Gastroenterology, who's published some guidelines on donor screening for fecal transplant, and I would say that generally speaking, we ask IND sponsors to follow those.
There's a question down here.
I'm David Challoner from the University of Florida. I had the great opportunity to chair the IOM's Committee on 510K. And so this is primarily, I think, a question for our FDA representative. Does 510K, the principles of engaged in that, which the IOM was very critical of, involved at all in the decisions, regulatory decisions that your division makes? And then second, what is the taxonomy, as you see it, of the corporate and industrial enterprise that is developing around this new product opportunity? I mean, for instance, when the IOM looked at devices, I was struck by, except for a few big players that were similar to Big Pharma, the device industry was all over the country in every small town, and every representative in the House of Representatives had a device lobbyist. What's building up around this?
Again, I'm not 100% certain I understand your question. Could you?
What's the business enterprise look like now that you're seeing out in the country as these products are beginning to come to you?
Well, again, much of that is confidential. We can't really speak about INDs that are under review. But you can go to clinicaltrials.gov and see what sorts of clinical trials are being done, and that generally gives a pretty good idea. I don't know if I can be more helpful than that.
You'd be surprised how much money people think they can make
Yeah.
just from poop.
Yeah.
There's a question up here.
Yeah, I'm Bob Calvin from the HRQ. We've heard throughout the day that over the course of the last century or so, we've evolved away from healthy things going on in the ecology of our guts. But actually, if you look at life expectancies, they've actually increased rather dramatically over the last century. In fact, if you look at other developed countries, they're increasing more rapidly here than they are in the U.S. How would you justify that?
Is this for me? I'm sorry, I'm having a hard time hearing. Could you repeat the question?
Yeah, I was just saying that we've heard throughout the day that we've done a lot of things to depart from the natural ecology of what we have in our guts. And it sounds like it's quite bad. Yet at the same time, over the same interval, life expectancies have increased rather dramatically, and they've increased even more rapidly in other developed countries. How would you square that?
Maybe I'll take a crack at that. Or Billy Joe can try. I don't think I have an answer for that. I'm sure that there are people from previous sessions who might be able to speak to that more eloquently. I don't know where we are along the spectrum of humanity in its fullest extent, and the arc of humanity, and how long people will live in the future, and whether getting rid of infectious diseases has given us a lot of extra years of life, but we're not quite there yet because we haven't optimized the microbiota, or maybe the microbiota is more optimized than we think. So that's a non-answer. I have no idea, but what I'm going to do is something unconventional, is to ask a question of the chair of the organizing committee and call him up to the microphone to see if he's willing to give a scientific answer. And this is something that I've heard from other people, so I was hoping to get his opinion. And that is that people often say, why don't we bank feces from an early age, the purest stuff that's never been hit with antibiotics, and that we can keep this in the freezer and go back to it if anything ever goes off the rails later in life. What you said earlier, which was very interesting, is that the community doesn't settle down until you get to some age, maybe 2 or 3 or 4 years old. When would you bank feces, or do you think that that's a fallacy and you should bank it anytime you feel good?
Well, it's come to that time of day. No, I have been known to have said that I had sometimes wished my parents had saved my poop instead of other things of mine from early in childhood. Appropriately stored, of course. No, it's an interesting question. So when does the system reach maturity? That's in part. And for what purpose might it then ultimately be intended? And that could, of course, be a big determinant of when it might have been best harvested. The ways in which the microbiome is being assessed for functional capacity, and actual realized function is somewhat crude right now. So when we say that those maturation curves, you saw the plots of the Malawians, the Amerindians, plateauing at age 3, Marty showed that. We had a study in 2007 that suggested even by age 1 in the US, there might be something akin to a mature-like picture. Well, we're using pretty crude measures. Jeff has shown that if you start looking at very specific families of genes that are responsible for either producing or metabolizing key vitamins, well, those things are sort of moving in divergent directions during that period of time, and they may not mature for some years later. And likewise, it may be that accumulated exposures to key environmental challenges must happen and only happen later in childhood, perhaps later, perhaps post-puberty, until you have a fully functionally mature microbiome that you'd most want at age whatever, 47, say, in my young case, or no, I wish. So I think it's hard to answer. I would just say I'd be talking about late childhood for sure, in part to know that some of these later effects of an altered microbiota from very early childhood hadn't already manifest themselves. So that's a long-winded way of saying late childhood, perhaps, and it depends on the context, and in many ways we don't know yet. Could I just add one,
Please.
just brief, because there was a really interesting question about, just prior, about this apparent...
Life expectancy.
Yeah, it's a dichotomy. It's an apparent contradiction in what you may have heard from today about degradation and loss and missing, and some almost dire predictions of what we might be doing to that which has been such an important part of us. And yet, on the flip side, obviously improving health metrics measured in various ways across longer periods of time. I think part of the answer to that dichotomy is that you're talking about forces that are operating on very different timescales. The adaptation of the microbiome to us has happened over tens of thousands of years, and certainly even more recently over hundreds of years. The kinds of forces we're talking about today are very short-term forces. We may not yet have seen what the consequences might be, other than in very obvious ways like the emergence of resistant organisms and resistant genes. But there's a lot of, some people have said plasticity, but also resilience, abilities to adapt and accommodate within the microbial communities. And I think maybe that's happened alongside the added benefits from these helpful interventions like vaccines and clean water and so forth. And we may not see the longer term consequences that are going to happen over a slower time scale of slow changes today.
All very interesting.
Thank
There's
you.
a gentleman down here who's been waiting.
Oh, who's next?
You.
Thank you.
Yes.
Henry Foster, Mayor Medical College. I just wanted to speak to, there was some ambivalence about the whole issue of life expectancy, what's happened over the past decades. As an obstetrician gynecologist, I have to remind people that the quickest way to increase life expectancy is to reduce infant mortality, since it's set at the time of birth. But if you want to get better definition, you must look at post-infant mortality, because you've eliminated the period of fetal death.
Good point. There are two gentlemen here. I'll go for the one in the back first.
Oliver Fine from Weill Cornell in Manhattan. I was wondering, is there any sense of what the price to the consumer is going to be of the fecal transplant? Has it gotten that far along that we have a sense of what it's going to cost, and what does it mean in terms of access to this therapy?
So we don't have any approved products yet. Currently, there's a group, and I don't think this is a secret because it's all on the internet, who is providing material for use. They are charging, but that is, as I understand it, cost recovery for screening. And if I recall correctly, it was about $500, but don't quote me on that. Now, what happens when you have an approved product and the market forces go to work? I couldn't predict.
I think it's a great question, and it will differ country-wise as well. So if you look at a country like Canada, where that kind of intervention, if it takes place in the hospital, would be covered through your healthcare system, it would be very different here. Access would differ globally, and I think that's an important question because I don't think we want to see that happen. I think we want to see access occur on an equal basis everywhere.
It's an interesting question in the context of the number of videos you can find on YouTube about this. So the fact that people have taken the initiative in some circles to do it themselves means that if the barrier is set too high, one wonders what's going to happen in the background. Question down here.
Gus Aguirre, University of Pennsylvania. We've heard a lot about the fecal transplants for changing the enteric microbiome. Yet from slides earlier this morning, it seemed like the oral cavity is far more complex in terms of the microbiome. Are there any potential translational applications for that?
So
I would say yes.
yeah, for the oral microbiome. So there are conditions that have been linked to a change in the composition of the oral community or problems there, and so cavities and periodontitis are two of the conditions that are known to be linked to specific colonists or pathobionts of the oral community. And there are people who have proposed trying to colonize kids with strains of Streptococcus mutans or relatives of Streptococcus mutans that are non-karyogenic. So that's an active thread in the oral microbiome community, although I don't know whether there are any products that have reached your desk.
Not that I can talk about,
Not that you can talk.
but I would add that something that hasn't come up at this meeting, but I think is extremely interesting, is the microbiome of skin, and you know the potential there for treating all kinds of things. So.
Yeah, there are things in the works on skin too, but as far as I know, nothing that has reached the clinic. The question here.
So the discussion is focused a lot around therapeutics, and it's clear with the inter-individual variability of the microbiota, how rapidly it responds to treatments or just changes over time, that diagnostics and monitoring is going to be incredibly important. And I was wondering if you could provide a thumbnail of what this looks like in the future. Is this simply just reading out species over time? Is it more complex than that? Are we going to have devices in our toilet that are telling us what's going on in our microbiota on a daily basis?
That's for you.
That's for me. That's a great question, and a question that is a difficult one to answer. I think we can all wonder about how we're going to monitor ourselves over time. I could imagine that cruise ship bathrooms will have a device to monitor to catch an outbreak before it becomes a system-level event. What I don't know is whether there are other problems that are specific enough that we'll know that we want to look out for them and nip them in the bud before they become a chronic health disease. Maybe I would turn this around to Scott and ask a question that plays off of a topic that came up in an earlier session from Stan Hazen. What if there came to be really strong proof for a causative link between bacteria that produce trimethylamine in the gut community and a risk of cardiovascular disease or a cardiovascular event? Would then you guys need to go back to already approved products and assess the risk that wasn't realized before that they might impose on patients? How is that going to work?
Well, that's a big what if. I don't know. I think that obviously you can't go back and rescind fecal transplants that have occurred, but I think it's like with any safety issue that comes up, you do your best going forward, and if that was shown to have a positive correlation, then that would probably be something that would be screened for in donor feces. But my understanding of the link with the conversion to trimethylamine to TMAO is that it's meat eaters. So that's a large section of the population.
That is a large section of the population. Okay, so I think I'm going to take that as the last point, and thank you all for participating and listening, and maybe give us all a big round of applause.